Hi, everybody. This is another one of our videos to get you prepared for our Animal Behavior Research Project Preliminary Data Analysis Lab. As part of the pre-lab, you were asked to try to organize your data in a way that would be conducive to doing statistics and graphing, and this is one of those videos to help you get that ready. This video is going to focus on organizing data for people who design their experiment to include focal sampling with continuous recording, noting the frequency of behaviors. Now, if you did design your experiment that way, you're in luck because this is actually one of the easier ways to organize data. So what we're going to do is walk through an example of student data where they did use that same experimental design protocol and think about how we can get that data organized nicely to make it easy to run your stats and produce your appropriate figure. So the first thing that we want to do is talk about the data that we're using for our example so that you know what you're looking at. And this data is available in our Carmen page in the modules tab under our lab resources module for the appropriate week. So let's take a look at what these students were doing. These students were interested in the effects of differing light environments on the movement rates of water striders. So what our students looked at were water striders in really high light condition tanks versus low light condition tanks. And they noted the frequency of both limb movements and positional movements. Positional movements meant moving the entire body, all 6 limbs of the water strider moving from one location to a new location. Limb movement is when the water strider was positionally stationary, but moved its individual limbs. So what our students did for each 30-minute observation session was select 3 different focal individuals, and they watched each of those focal individuals for 10 minutes at a time. You can see that noted on their observation sheet. So they have the group member who did the observation, the date they made their observations, and each of the 3 focal individuals that they watched for a given 10-minute period. For each focal individual, they made note of every single time there was a limb or positional movement. Now, this data is actually pretty well organized to start with. They've got all of their high light tank observations organized and all of their low light tank observations organized together. If your data is a little bit more individualized, as in you don't have one big data table where all the group members' data is included for each one of those different variables, that's okay. All you'll need to do is copy and paste those individual observations into one big consolidated table. If you actually watch the other recording where we did focal sampling with continuous recording, but for durations of behaviors, there's actually a nice example of how to go from individual group member data tables into one big compiled data table. So you could watch the beginning of that video to get an example of how to organize data in that way if you'd like to. Okay, so what we've got here is all of our data for the highlight tank versus all of the data for the low light tank. What we're going to do is compile this into one big data table to make things nice and organized. So what I've done is taken our unorganized raw data tab, and all I've done is copy and paste over those basic things into our organized data tab. The only change that I initially made in copying and pasting this over is that I've just gotten rid of the group member who made the observations because we don't care about that, and the date that they made those observations because we don't care about that either. So at this point, I've got the exact same data tables. The only thing that I've done is delete out those individual values. Now, the only real change that we want to make to this is instead of having a separate data table for our highlight versus low light tanks, we're just going to combine those into one big old data table. So what I'm going to do is copy and paste over the highlights and say Control C, and I'm going to shift that over there. I'm going to do a little bit of zhuzhing around here with my columns so that I can read those headers more easily. There we go. So the movement is all one word and positional is all one word, easy. And then what I'm going to do is copy over everything from my low light tank. And notice I don't need to recopy over those headers because there's just no need to do that. Now, the way that these students have this organized is they've got one big old merge cell for high and low, and that is generally fine. I prefer to have each individual row labeled. That is personal preference. So what I'm going to do is type the word high here, and then just drag that down to encompass all of my highlight observations. And then I'm going to write low here and just drag that down to encompass all of my low light observations. Easy peasy. And then at that point, I can delete this. Here we go. And label it light conditions, which is more reflective of the actual independent variable that I'm interested in rather than tank. So at this point, I've got all my data organized really nicely so that I can easily calculate out those averages, standard errors, standard deviations, variances, run my t-tests, and get things plotted out in a bar graph nice and easily. So like I said, this is a pretty easy form of data to organize. The only tripping point you might run into is that if you don't already have things compiled according to independent variable, then just go ahead and watch that other focal continuous video that focuses on duration data, because it's got a nice example of how to compile individual observations into one nice big spreadsheet. Otherwise, you should be good to go. Have a good day, everyone, and good luck with your data analysis.